Hi, now let's talk about stem cells and cell death. So the cells, from the beginning, the cells will differentiate into a terminal state, which is a specialized cells. And cell differentiation is a process of cellular specialization in function during an organ development or tissue repair and even normal cell turnover with the same genetic material. Especially quite recently, stem cells become of great interest to biomedical engineers. And stem cells are unspecialized precursor cells that can differentiate into a diverse specialized cell types, or it can self-renew. So for example, in developing embryo, stem cells can differentiate into 4 specialized cells. So that's great. And in adult organisms, like us, we are already adults. What are our stem cells? And stem cells acting as a repair system. So we know that we got some injuries, small cut, in a few days we will heal this, and over time it will even like disappear. So for our body and maintain our normal turnover of regenerative organs, such as blood, skin, or intestinal tissues. And stem cells are found in all multicellular organisms. So for us, like biomedical engineering, even out of curiosity, we are curious about how like our blood are keep going. And for example, red blood cell in our human body, they actually last for only 120 days, and then they will die. So we have to keep generating those new red blood cells inside our body. In fact, it's in the bone marrow. So this potential use of stem cell as a repopulating and tissue regeneration is of a great interest to us, especially when there's injury, there's an accident, a loss of tissue, or a problem in organ for replacement. So potential cell source as a stem cell is very important, and cell replacement therapy for diseased tissue, also for creating an engineered tissue, such as, let's say, skin substrate, skin replacement for burn patient. And as a seed cell, stem cell would be very interesting to us. So let's consider about the definition of stem cell. So last time we learned that in vivo fertilized egg and that zygote has the ability to become the whole cell types in a complete organism. So that we call as a totipotent stem cell. And once they divide into, let's say, for example, 8-cell embryo, and they will start to make a sac, which is called a blastula, an animal embryo at the early stage of development when it's hollow ball of cells. And that blastula now contains a small inner cell mass, and that is called the blastocyst. So it's a mammalian blastula in which some differentiation of cell has occurred. And that inner cell mass can become many types of the cells in our body. So we call it as a pluripotent stem cell that can be cultured and that can be differentiated into neural cells, that can become cardiac cells, that can become blood cells. Of course, there's a more differentiation so that particular levels of stem cell, which can only differentiate into one type of terminal cells, is called unipotent. So these term, toti, comes from Latin, the whole, and pluri, you remember that in English, a single versus a plural form. So that pluri means in Latin, very many. And this potency means having power to become any kind of cell types. So this potency definition of stem cells can be explained as a first, the stem cell which can construct a complete viable organism is called totipotent stem cell, or some people say this is omnipotent. So good example is a fertilized egg. And descendants of these totipotent stem cells, which can differentiate into almost any cells in our body, is called pluripotent stem cell. A good example is that embryonic stem cell. Embryonic stem cell, however, is coming from the placenta, so it cannot form placenta. Now, these pluripotent stem cells can and downwards, the stem cells can differentiate into a number of cells, but only those closely related family of cells we call as multipotent stem cell. A good example is hematopoietic stem cell, which is a blood cell making stem cell. And then the last part of the stem cell is the stem cell can differentiate only into one type, such as muscle cell or skin cell. So there exist these stem cells which can replenish our skin or a muscle. So that's called, because it can differentiate into only one type, it's called a unipotent stem cell. And now let's discuss what kind of stem cells are available. So first, isolated from an embryo, the most potent stem cell is called embryonic stem cell. But as you can imagine, embryonic stem cell is after this fertilized egg. So from the religious point of view, this can cause an ethical issue for stem cell research. So there's another breakthrough happens. We found an adult stem cell, which is from the tissue of an adult organism, such as you can take out a little bit of the skin of myself, and that if you can make a stem cell out of this, that can become called stem cell. Or for example, bone marrow or peripheral blood, and it contains stem cell, and that's called adult stem cell. Now, a revolution in the stem cell field happened not more than 10 years ago, a decade ago. It's called IPS, or induced pluripotent stem cell. By definition, induced and pluripotent is a very much capable, potent stem cell. And these are artificially derived from a non-pluripotent cell, typically adult, body cell. So it's an adult somatic cell. And that, that gave rise to a Nobel Prize in 2012. Do you know when the first paper on this came out? Majorly by Shinya Yamanaka from Japan. The paper in Cell was published in 2006. So in only about 6 years, he got Nobel Prize out of this discovery. So that means a lot, that is how important this field and how interesting this field is, because you can take out adult somatic cell and reprogram, means he has 4 factors of transcription factors into the cell and reprogram it to a pluripotent stem cell. So that resolves the ethical dilemma using embryonic stem cell. And so this is the wording in the Nobel Prize for the discovery that mature cells can be reprogrammed to become pluripotent. And you can ask questions like, okay, what are the stem cells? They are cells, so they may look similar. It's hard to find. So by definition, they exist in very small quantities. That's why it has been so long to discover the stem cell within tissues of all adult organs. So let's consider the characteristics of stem cell differentiation. So you look at this stem cell, which is quite interesting. Stem cells were divided into 2, and it can give rise to 2 stem cells, but it also possesses very interesting feature. So you see stem cells divide into the same one, so the self, and you look at this, it's just a cartoon, but it's a different one called committed progenitor cell. So it can divide into the same one, and that will give rise to more and more stem cells. That's called self-renewal. While this also gives rise to progenitor cell. What's progenitor cell? Means a cell which has a tendency to differentiate into a specific type of cell. And it's a push it to differentiate into that target cell. So you can look at these progenitor cells, then they will amplify. So it is called transit amplification stage, and then eventually they will become a terminally differentiated cell. So this very interesting property of the stem cell characteristic is divided into here. First, asymmetric cell division. And this part can self-renew for forever, in fact. So that's called limitless self-renewal, which is quite interesting. And also this multi-lineage of differentiation. And so all this property allows the stem cell to repopulate of tissues in the recipient or in our adult. So I want to bring an example to explain stem cells, especially hematopoietic stem cell differentiation. So it's a little difficult term, but hematopoiesis is a hema means a Greek blood, and this poiesis is to make. So think about our blood. Our blood contains red blood cell, which is almost composing like 40 to 50% of blood volume. However, there are also white blood cells, which has a variety of cell types, and also a platelets. So where do they come from and how are they made? And actually, there's an active process of hematopoiesis, or there exists hematopoietic stem cells, usually inside our bone. So stem cells can become divided into, let's say, lymphoid stem cell, and lymphoid means lymph nodes. So it's related to our immune system, and that lymphoid stem cells resides in thymus. That's a gland underneath our chest, and also it's related to a spleen, which is responsible for a lot for immune function and also filtering old senescent red blood cell. So that become a lymphoid blast, and that terminally differentiate into B cell and T cell, or we call B lymphocyte and T lymphocyte, which are part of our white blood cell. And the other lineage is going to myeloid stem cell. Then myeloid is coming from the term bone marrow, so inside our bone. So myeloid stem cell become myeloid blast, and it will differentiate into a monocyte or granulocyte. And monocyte is a precursor for macrophage. You know, macrophage is the name meaning that big eater. So it eats up foreign molecules or maybe dying cells around. So these are a good immune function. And also these granulocytes are composing of various kinds of white blood cells, including neutrophils. And the erythroblast becomes a red blood cell, and megakaryocyte blast actually pinch off from the cell into making plate cell. And this red blood cell or RBC and platelets having no nucleus. So in a nutshell, hematopoietic stem cell can differentiate to these various kinds of blood composing cells. So it's called multipotent stem cell. So where are the stem cells located? In the stem cell field, people now consider how the stem cells resides and interact with its microenvironment. So maybe it's not, let's say you want to create a living, functioning heart from stem cell. Does that just growing in vitro, the stem cells will form a heart? It might not be true, because stem cells needs a proper environment to differentiate properly, so that it needs a nearby cells and environment to interact with. So that concept is coming as a stem cell niche. So there's a good review. So this is a reference, so it's a little advanced, but I think it's important to understand the adult stem cell locations in our body, and that's termed as a stem cell niche. Okay, so niche, it's referring to a microenvironment within the specific anatomic location where stem cells are found, which interact with stem cells to regulate the cell fate. Just for example, like the thymus, that T cell in the immune system, T comes from thymus. So to develop into a properly functioning a specific T lymphocyte or T cell, they have to mature inside the thymus, and that environment provides a stem cell niche. So let's consider like tissue type, a hematopoietic system. That stem cell is inside the bone marrow. And another example of a interfollicular epidermis are in the basal layer of epidermis. So this is an example of a stem cell potential use. You can see many diseases or baldness, blindness, missing teeth, wound healing, virtually almost all kinds of disease may benefit from understanding and engineering of this stem cell. So that's why it's so interesting. Another aspect of a cell is not only proliferating, but also cell death. That part is also very important for us to understand. And do cells commit suicide or homicide? That's what I put it this way. So there's a very interesting phenomenon that, let's say, the development of our finger or web. So we originally developed an embryo with like this. And then the cells in between the digits, they will start to die out this red colored one. And that makes in terms of finally these fingers and during development. So this is a very precisely programmed ways of death so that they can make our embryonic limbs. So that is an important finding for us, and that happened. This programmed cell death is called apoptosis. So the cells can die in different ways. So that's I commit suicide or homicide. So this corresponds to suicide. So that cells dehydrate, cells shrink, and chromatin that including inside the nucleus will condensate, and you see making a nice blob and then chopping off called apoptotic bodies, and they will be uptaken by cell-eating cells nearby, such as macrophages. And that's called cell-eating, meaning phago is eating cytosis. So phagocytosis by macrophage or nearby cells can clean up. And so what's important here is that membranes are intact. So membrane integrity is preserved to a very clean way of cell death. On the other hand, let's say you've got a mechanical trauma, you got hit, and then you have a bruise. And what happens in that case is cells premature death will occur from this injury. So the cell and mitochondria will swell, and eventually it will burst out. So its content will be exposed outside. That will cause an inflammation. And that inflammation makes you see the redness of the tissue and the swelling of the tissue and also the pain. This process of cell death, or I named it as a cell homicide, is called necrosis. So our tissue to be maintained, it has to proliferate. At the same time, it has to also die. So that balance of proliferation minus cell death will make our tissue maintain. So apoptosis is called a programmed cell death or cell suicide. And that, because we name it programmed, means it has a very precise sequence of chromatin condensation, cell shrinkage, pinching off, and forming apoptotic bodies, and leading to no or little inflammatory responses. While necrosis is kind of cell murder because it forms a fatal immature cell injury and death. So premature death of cells and living tissue due to the injury or trauma or disease such as infection or failure of blood supply, such as ischemia. And cells swell and burst, releasing their content and often triggering inflammatory response. So I brought like two images which has a more detailed information here. So comparing this necrosis and apoptosis, I want you to read through because this is such an interesting and important. And another picture here is made out of quantitative phase microscope, which is great in terms of you don't put any external labels or contrast agent, just based on optical refractive index change. The thickness of the cell is color coded, and you see from top to bottom how these cells are getting decomposed with a small microblebs and disappear. So this is a process of apoptosis, which is now possible to look at it without any labor, a very intact process of apoptosis. So next time, we will talk about cell culture technologies. Thank you for your listening.